Okay, so I will present a study that we are still carrying on. So we've been doing that for over 2 years, and we are doing a long-term study of what we call stem cell derived osteocytes, and we have created a bone-on-chip in order to mechanically stimulate them. The end goal of this study is to have some contribution to the improvement of the design of a large bone defect solution, in particular in the case of cancer resection, as Hervé mentioned it. So when you think about organ on chip, you're trying to recreate an organ and keep it alive, and be as close as possible to the in vivo situation so that you can compare, or at least have some aspect of the functionality of the system that you have created eventually to implant it. So you have two views. Either you go fully synthetic or you go fully graft. You take a completely organic graft, and then you have a system in between that use either just a scaffold or a scaffold and a cell, and the scaffold can be synthetic, or nowadays more and more study have been done, and especially for small organ and small animals, with the decellularization of the whole organ or the tissue, the recellularization, and then the implantation. And these systems are very interesting in order to use them for an organ on chip, for instance. In some organ, when the structure is extremely simple, you can do a full bottom-up. So in an organ, you can dissociate the parenchyma and the stroma, and for bone, most of the time we study the mechanics of it, we study the formation of it, so we study the stroma. So the first family of cells, of course, it's not the whole organ, but the first family of cell we started to look at was the osteocyte, because there was a key of the remodeling process, and also they have a very peculiar structure, and they were interesting to study. So we started from mesenchymal stem cell because they have clinical relevance, but we could have also gone for primary cell. At the time we started the study, human primary cell were not isolated yet, so we started from this one. So we had to do a proof of concept. So the first study was to figure if we can decellularize tissue, have it in a form that it is reseedable, and then reseed cell on it and see if they would survive. So that's what we did. In order to look at that, we did the decellularization process, the recellularization, and then we looked at them under confocal, under different wavelengths. So this is just autofluorescence, and we saw at the time there were very few surviving, that some survive on the edge attached to some pieces that we could see extremely well in red. This primary study was also useful because, in fact, we realized that we could isolate the morphology of native osteocyte from the red wavelengths. So we used it on another study, which I will show, which is on the cell reconstruction for the model. In any case, we had a system that we could use to do a bone-on-chip in order to study the in situ mechanobiology in 2 cases, either in healthy or, diseases, it's just a fracture, and to assess some aspect of the functionality of the construct. So the parallel study is that we have an experiment which is our surrogate organ on a dish, and on the other side we have the model. The model, we use it in order to have precisely the mechanical stimulation at the tissue level and at the cell level with as much precision as we can, so in explicit modeling. So why do we do that? The first thing is to understand how the tissue work, what are the native stimulation in the case of healthy or fractured tissue. We know we're going to face major challenges. The first one is that it's a completely multi-scale system. The second thing is that it's also a multi-physic. From this, hopefully, we can have some insight on how we can improve tissue engineering. So the scale that we're looking at is a tissue scale, the subtissue scale, so the osteonal structure, the subosteonal structure, so the lamellar structure, and then finally the cell, and the cell will also decompose it. So first we did our bone on chip. So what we did, we took a piece of bone from a man and we decellularized it and recellularized it. We had two ways because at the time of the study we didn't know the direction it will take. We didn't know if the MSC would take the osteogenic direction. So we had on the side another model that was using MLO. We could have taken ICGSW3, but so we had the MLO, which are early osteocyte. We reseeded them, that was kind of our osteocyte model. And then on the other side, we had the MSCs, and we were hoping they would become osteoblast and eventually osteocyte. And so that's what we obtained after many, many months. So we are looking at a bone which is potentially cracked, so we have a little crack in the model. So first for the decellularization took us about a year to figure it out and to do multiple baths of disinfection, and then we placed back the cell. We created our little basically 3-bond bending test, which has two interesting regions. The first one is that we have a small crack which stimulates a small fracture where the cell will populate and create a new sort of newly formed tissue. And then we have the compressive site that could be our native compression, and we do that in different directions, but I will show the longitudinal because that's the one we started with. And then for the different aspect of the multiphysics, we're really looking at two only right now, the mechanics, so the interaction between the cell and the substrate, because we're stimulating the substrate. The cells are stimulated by the substrate and not by flow. And then we have the chemical environment, we're looking at the chemical release. So for the cell morphology, basically in the compressive, we have recreated a native osteocyte morphology, and for the newly formed bone, we have the morphology of the new cell, but I will only show you a study on the cell body because those reconstructions are very time-consuming. So for the cell observation, after a few days, we realized that our cells were living very well. They actually were multiplying and surviving. The other aspect that was interesting is that they're reorganizing in vitro as in vivo. The MLO were trying to recreate some sort of osteonal distribution on the lamella and staying on the substrate, and the MSCs were going inside the aversion canal. Now, after many months, we are looking at the survival of our cell. We can only go down to 80 micron, that the capability of the confocal. We know we have cell as deep as 80 micron within the within the bone. And when we're looking at histological cuts, we can see that the cell really reorganize as a osteon, and they leave a print on the bone. What's interesting too is that they stabilize. We have a stable population and we do not have an overgrowth of cell. Now for the MSC study, which is the more relevant one for us, we are looking at the morphology and in the newly formed bone, so in the cracked region, and this is done without cyclic stimulation, only we have spurious stimulation. we can see that they do recreate some sort of network, and they produce process, and they have several process. And we have also on the edge, really on the edge toward the exterior, we have longer cell that looks like bone lining cells. Now, we had to characterize them in situ. We have not done DNA analysis yet, so within this fracture, those are my white line, we have looked at several markers. So first one is the viability, so they do survive. We looked at one wavelengths that could correspond to hydroxyapatite, so that would be the yellow dot. For the purple dot, that would be one marker, which is E11, and the red one is sclerostin. And so it doesn't show very well, but we have a few examples of that. We do see that some cells express E11 and sclerostin, some more just sclerostin, some more just E11. We have an heterogeneous population, but we have at least few osteocytes within it. Now for the modeling, because down the road we want to know the mechanical field around the cell, we first did the tissue analysis, so it's a hierarchical model. We start with a CT scan of the bone on chip, we segment it, and we recreate the osteonal structure. We know from previous study in 2D that we have done that we need to take into account three main points. The first one is the anisotropy and the heterogeneity of the tissue. The second one is that we have an heterogeneity of the cohesive strength, which is due to the bridge fibers that remain within the opening or at the interface of the cement line, and then we have an heterogeneous diffuse damage. And so we took this into account the best we could, and in 2D we were able to reproduce the behavior of, the numerical model is able to reproduce the behavior of the experiment. Now in 3D, we are struggling because it's 3D. So we do have the heterogeneity and the cohesive interface in explicit form. We're still working on the anisotropic diffuse damage. We do have the model, but it's a challenge to implement it. Another point which has been observed by schedulers is that depending on the usual load that the cells are subjected to, they tend to produce osteons which have a specific collagen orientation. So that's something we are going to look down the road, but we can't affirm yet. Right now we basically have the tissue produced when you have very little stimulation. So looking at the field within the tissue, so we do see that the cement line is kind of critical because it really isolates what's going on within the lamella, so for the active osteocyte from the interstitial bone. Now we are looking at the mechanobiology, so we have the mechanics on one side and the chemical release on the other side, which is the red here. And that would be calcium orange bars. We're looking at the calcium release. And then from the field that we have, so we're doing basically loading that we remain within the elastic regime, is just a slight walk, and we are transferring this stress field at the cell level. Why? Because experimentally we have observed that when we load the tissue, we have a diffuse damage that occur. We have first a debanding of the lamella and then microfracture that develop within the lamella and that directly interact with the cell process. So two things are important here: having a reliable tissue model. Second thing is having a reliable cell morphology. So for the cell morphology, another struggle. So we started with confocal imaging. Why? Because we want to look at them alive. You can have beautiful reconstruction with synchrotron, but you cannot easily put your bone on chip in a synchrotron. So we did a lot of image processing in order from very poor confocal image have a reconstruction that could approach a synchrotron. So we did our cell segmentation. So it's a simple parallel plane segmentation, the body reconstruction, and then for the process we have a few cases. You can end up with a case where you actually do not have the connection fully segmented in the image, so then we had to find little way to reconnect them. You have the branching, and you also have a lot of noise. So first we have a noise regulation, so we smooth a little bit the response, and even though with that we still have, because we only get the central axis of the processes, we still have problems due to the diameter of the pericellular matrix, and so we have further smoothing, which is shown there, and then we have the connection. So I'm going to show mostly what my student, who was very dedicated, she's called Avila Gassi, and she worked mostly on the body, and you have the reconstruction of the processes, but I won't show you the mechanics yet. So we know exactly where our osteocyte was. So we, instead of having to load it in every possible direction, we actually know what is the usual direction when you're walking of the specific osteocyte that we're looking at. And we are extracting the morphology, adding the pericellular matrix and the matrix, and for the matrix we want to overlay all the mechanics that we have done in the past. Now, two cases. The first one will be healthy. So the first one is fractured tissue. So the newly formed tissue, which is more isotropic, much softer. I will show you, but still lamellar. So we'll still observe alternation of collagen orientation and sort of surrogate lamella. They're a little bit thicker than within a lamella, but the tissue that we observe, especially on the edge, resemble the circumferential lamella that you have that separate the bone from the bone marrow. So we looked at it, we did different type of histology to have a grasp of the structure of that tissue. Sometimes we observe very interesting things. They can do blobs, and within the blobs, so in the yellow, we are looking at the hydroxyapatite, at the calcium crystal deposition that we also marked in other type of histology, and we know that we have collagen 1, and you have the cell, so some clusters are more important than others. Now, the global mechanics of that tissue, we obtained it by, so that's our system where you have the bone, the cell, and the newly formed tissue. And in the red would be basically the response of the bone, and we do feel it that it is the bone, so that would be the red curve. And then after some point it actually cracks, so it cracked really sharply because we're kind of unstable. It's a 3-point bending, but that's all we can do with the little thickness of bone we can have in a cortical of a human person. And then we were actually loading the newly formed tissue, which is much more viscoelastic, and it's about a quarter strength of the native bone. So now going for the model, so you have the chemistry on one side and the model on the other side. So the tissue field, we can have an idea of the range the cells are subjected to when we load them inside this bone-on chip, which is to represent basically if you were walking on a site where you had a fracture. And we are pulling with a few, so I think we're less than 100 piconewton on the cell, and we're looking at the response. And on the other side, we have the experiment to measure the calcium release. Now the other side would be the walking person. So the walking person right now is more theoretical. It's the extracted microstructure of the native cell that we embed inside the native tissue. but we're doing some twist on the native tissue because we're looking at the effect of anisotropy. So we have two cases, the isotropic that would be your average bone property. These studies have been done by other groups, especially in Ireland and in England. And the other side is looking at the anisotropy. And people have not really paid attention to the anisotropy around the cell field right now. So on the top you have the isotropic case, and on the bottom the anisotropic case. We have a system where we have a cell with a nucleus. We took a nucleus site from our image, but also from people who have isolated primary osteocyte. We embed it into the model, and then we can also look at the effect of the PCM. So the PCM is this thin layer of protein which is around the cell. It's much softer than the bone, but it kind of protects the cell from a direct contact and direct stimulation, mechanical stimulation from the matrix. What is interesting is with an anisotropic matrix, the distribution of the field is very different. You have about a factor 2 of less load on the cell, we're still in the elastic region because we're assuming the person is walking normally, but if we were in the case of the person is running, for instance, or jumping, we would have then the creation of new damage, and that would be interesting to see the interaction with the cell processes. Now, for the sort of molecular signaling, it's not the molecular signaling, it's more the chemical release. So we did some experiments. So that was at the beginning of their life. So only a few days. We started like this, where we have sort of known population, MSCs on one side and MLO on the other. And we are, so it's, I said it's a sort of single loading case where we load them for about half an hour to an hour, and we're looking at their response. What we observe is two clear, distinct response, which has been observed by other researchers using fluid flow stimulation. Here we stimulate them by direct mechanical loading on the substrate. And the MSCs tend to sort of lose their cytoplasmic calcium after a very small amount of load, while the MLO sustain their calcium. Now, the dye we used were not really calcium specific, but we did a calcium specific afterwards. Now, we knew also about the load the cell were exposed to, and looking at the shape, because we have the image of the shape, we can do some rough calculation of the local load on the cell to the chemical amount of calcium that they're releasing. So the goal here is to have basically sort of a relationship. Of course, it's going to be a simple one, but where we have the amount of calcium variation versus the amount of local load, so being measured as precisely as possible. And that allows us to end up to this type of chart. So you have the mechanical analysis on one side and the chemical analysis and characterization of the cell differentiation state on the other side. And in the middle, we can correlate the amount of calcium that we have observed. So the variation is measured from the variation of the fluorescence, and on the other side, we have the variation of the force. So on the top, you have the osteocyte, which basically sustain their calcium. Now, retrospectively, it makes sense because it seems that the cell basically adapt their amount of calcium to, their calcium cycle to the amount of load they expect to see. So the osteocytes are within the matrix, which is extremely stiff, and they're going to be subjective to higher load, while the MSCs are usually protected within the canal or the little location, and they're subjective to less load, so they would lose it faster. And then we have later on a mixed population, so we have strange curves and a cell that recover and do not recover it. So that would be it. So I will thank people who are helping. So Eric Schmidt is the person that helped me to build the device, modify the device, and adapt them to all types of load. And then we have a team of imaging at LBPA in Cachan, and other students and engineers who have helped at LMT. And of course Murad, who helps us with the cell and the reseeding, and people who have given us money, and it started with the funding of NSF for this reseeding proof of concept. So I will thank you, and I'll take the question first. Yes.
What's the importance to have access to exact morphology of those osteocytes? In which respect the exact morphology of the osteocyte affects the conclusions of your study?
Everything. So this comes from previous study that we have done on bone, where initially when I started to work on bone, I was working with Monte Carlo models. And we started with very simple geometries, so basically tubular fibers, circular, cylindrical fibers. And we realized, and we compared this, that people were finding all kind of relationship with the load and the properties. And okay, so we looked at it. We were finding those relationships where we had this type of geometry. Then we started to make it more complex. We had more complex fibers, interpenetrating fibers, and then we went directly explicit. And at this point, no relationship that we're finding in the Monte Carlo ideal system existed in the natural system. So we started to work on explicit tissue. Also because you can see that the interface usually have multiple level of heterogeneity. Like for instance, the osteon is not only the shape, it's also all the wrinkle around it, it's all the layers. And for the cell, what's crucial is that they do create those processes. And these processes are embedded within the lamella. And when you look at a cell and where it is within an osteon, it's never randomly. And they're not right in the middle of a lamella, they're actually slightly on the edge usually. And so we think that there is an interaction with that. And the way they generate their process right now, the process that we have observed are under basically almost no load, but we want to do more cyclic now. But we think that the shape of the process will depend on the load, because the orientation of the collagen fiber they will secrete will depend on the load also. And so if you want to have the information at the level of the process, you need to have the properties the tissue it's interacting with and the shape of the cell, because the process are not exactly tubes either. They usually have small teeth which attach them along the canaliculi, and they do have small wrinkle, and they probably have a reason why it's like this, and the spacing of the teeth and etc. So for the shape is key. Other input that we have seen is when we were comparing, so that was when we were working on the cracking itself. figuring if we take into account the lamellar orientation, not the lamellar orientation, or if we take a generic fracture law. We had two parameters to take into account. The first one was mineralization, the second one was collagen orientation. So the mineralization, curiously, doesn't contribute much because it doesn't vary much, and somebody mentioned it, so we're within 15%. Now for the shape, again, the collagen orientation, we had a 350% difference depending on the collagen orientation to relate it to the fracture opening and the cohesive strength. So if we do not take this information locally, basically we would have not have a reliable fracture initiation and distribution. So my experience is that it's usually better in biology to have simple model, very complex shape, rather than very complex model versus very simple shape. You're usually much more off that way than if you have at least the geometry.
But how many degrees of freedom?
Okay, so the tissue one, we are at 320 million, something like that. In the cell, we're also over 6 million, I think.
How much time is it?
It's long. So the tissue itself, I think sometimes we had to run it for 2 weeks. But also we're not in optimum computational situation. So I'm sure it could be faster on a better machine. And for the cell, it takes about half a day. Which is okay, because from where I come from, if it runs for a month, it's okay. So it's just depend of your culture. Some people want super fast computation, and some people are okay with long ones. So.
I'm interested in experimental elimination of mechanical characteristics of single cells.
Yes.
Can you tell us something about that?
So you're thinking about nano-indentation, for instance, or things like that on the cell?
Let's ask about the method.
No, so right now, yeah, I would have loved to do that, but you have environmental AFM. Right now, I don't have access to that. So what we did is that we looked at the work by Adachi, who has done that, and he has different properties depending of how far you are from the process and on the cell body, and we used those for the cell property. Yeah, but yeah, that would be nice to do AFM. Yes. Yeah, well, another one here that we could do would be to load and do digital volume correlation. That would be another one. But it would be more rough, I think, more average. Yeah.
What about nanoparticles which are saying?
Yeah, you can do that. I've done that with cardiac cells. So I've done that with cardiac cells in gels with particles, and what we did is that we textured the gel with nanoparticle, and then we did digital correlation on it. Yeah.
Okay, any other question? I have a very short question, maybe a remark. At which, and it follows from Vittorio's question, how much detail do we need to know about the morphology? Where can I go? Maybe never. Okay. So,
Well, as time goes, you can do more because computers have more capabilities. I started with 2D, and with 2D we were doing explicit. My student who did that, he worked on it for 5 years. But in the US you can do that. Here for 3D, originally I wanted to do a few osteons, except my student was American and she did, we're going to do 64. So we did 64. I don't think you need that many. You can start with one and just focus on that one. And for the cell, you can start with one. Like right now, I'm downsizing a bit but I think it's good to have as much as you can on your model.
but we got any evidence that classical elasticity is good for describing this very small object?
Right now, we don't have just elasticity. We have contact, we have anisotropy, and we have damage. So it's not just, and we have different type of cohesive interaction. So yes, the parts sometimes are elastic, but we improve them as much as we can. The problem is that when you complexify your model, the time of computation increases as well. So if you want just a quick info, you can go for elasticity first, but yeah, then improve.
Thank you.